Hello everyone, the topic we chose was DNA replication. This process is really important because it ensures that each resulting cell will have a complete set of DNA molecules. DNA replication occurs in the nucleus during meiosis and mitosis. If our DNA didn't replicate, we wouldn't have enough gene instructions for the making of proteins. Basically, if our DNA didn't replicate, we wouldn't be here. In the model that we made, the purple pipe cleaner stands for the deoxyribose sugar. The pieces of straw stand for phosphate. The orange pipe cleaner stands for the base thymine. The yellow pipe cleaner stands for the base adenine. The green pipe cleaner stands for the base guanine. The red pipe cleaner stands for the base cytosine. The piece of cereal stands for the single-stranded binding proteins, also known as SSB. The little green square stands for primase. The orange little paper stands for ligase. The pink circle stands for DNA polymerase. The red bent paper clip stands for the enzyme helicase. It all starts with the DNA. The DNA gets unwinded by helicase enzymes. The unzipping occurs when the hydrogen bonds between the base pairs are broken and the two strands of the DNA unwind. This starts to create a replication fork, which is where the separation is taking place. The two strands of DNA will be used as a template for the attachment of complementary bases. The SSB proteins bind to the DNA strands to keep them separated. Then the primase comes along and indicates the DNA polymerase where to start. Then DNA polymerase goes along the strand, adding new nucleotides to the new chain continuously all the way down the molecule, following the base pair rules. The top strand is called the leading strand because it goes in the direction of 5' to 3'. The bottom strand is called the lagging strand because it goes in the direction of 3' to 5'. This is because the leading strand and the lagging strand are anti-parallel. The 5' and the 3' indicate the position of the carbons in the DNA's backbone. The 5' ends with phosphate and the 3' ends with an OH. DNA polymerase always goes from 5' to 3' because it only adds nucleotides to the 3' ends of the molecule, thus advancing to that direction. So then the primase has to place more primers so that the DNA polymerase can continue to work in the correct direction, which is 5' to 3' on the new strand. Then all of the little fragments get joined up by a final enzyme called ligase. The fragments that aren't fully made nucleotides in the lagging strand are called Okazaki fragments. They are named after a Japanese molecular biologist. The DNA polymerase keeps making nucleotides until it is done with the whole DNA molecule, which is of course way longer than this one. Next, the DNA polymerase proofreads each new DNA strand, helping to maximize the odds that each molecule is a perfect copy of the original DNA. Finally, the DNA ligase will join the DNA fragments together. Voila! And there you have it, 2 new DNA molecules. This process is also called semi-conservative because the 2 DNA molecules that were made consist of 1 old strand and 1 new strand. The old strands are also called the parent strands, and the new strands are also called the daughter strands.